Hi everyone, it's Dr. LeBlanc with Unit 14A, Bacterial Genetics and Conjugation. Our learning goals are to understand the mechanisms of genetic transfer between bacterial cells, to understand conjugation, to understand the difference between an episome and a plasmid, and to understand how the genotype of a bacterial cell can be changed using F episomes. This is an electron micrograph of bacterial cells in the process of conjugation, and you can see that there's a bridge, a cytoplasmic bridge that connects two bacteria cells. This is the bridge that the genetic information is going to travel through from one cell to another. Bacteria are usually haploid, which means that it's easier to identify loss of function mutations because they are not masked by dominant genes as they are in diploid organisms. A single chromosome is the only chromosome that exists in bacterial cells. They have a much smaller genome than eukaryotes. They can have plasmids, and they can transform or change genotype easily in the lab. They reproduce asexually. However, they can transfer genetic information from one cell to another, and that is the process of conjugation. Transfer of genetic material from one bacterium to another can occur in three ways. Conjugation, and that's the topic of this lecture, that involves direct transfer from one cell to another. Transduction, in which viruses transfer DNA between bacteria cells, and transformation, which involves the uptake of DNA from the environment, and that's something we do in the laboratory using plasmids. We're going to talk about conjugation. So here's a diagram of what happens during conjugation. You have a donor cell and a recipient cell, and both cells have their own chromosome, their own E. coli. If it's an E. coli cell, they have their own E. coli chromosome, but they can also harbor other smaller pieces of DNA, circular pieces of DNA, that can be easily transferred from one cell to the other when a bridge forms between the two cells. Josh Lederberg and Ed Tatum discovered genetic transfer in bacteria in 1946. They used Escherichia coli, E. coli, and they used two different types of strains. One strain were auxotrophs. Auxotrophs are strains of E. coli that are mutant and can't synthesize a specific needed nutrient. Prototrophs, on the other hand, make all the nutrients that they need from basic components like carbon and sugar and lipids. So the two auxotrophic strains that they used were one that requires biotin and methionine, but produces threonine, leucine, and thymine, and then another auxotrophic strain that produces biotin and thymine but needs to be supplied with threonine, leucine, and thymine in the media that they're growing on. So these strains can be designated bio minus, met minus, 3 plus, Lu plus, and Thi plus, and the reciprocal for the other strain. They asked a simple question: can one strain pass on its traits to the other? So they used three different cultures of bacteria. One of them had the first strain, which was met minus, bio minus, threonine plus, leucine plus, and Thi plus, and the other was the reciprocal. And they mixed them together into one tube. So these were the controls, the strains, auxotrophic strains alone, and this was the mix of the two auxotrophic strains, and they wanted to know, can we make a prototrophic strain out of these two? They plated the same amount of cells from each of the culture, 10 to the 8th cells, onto nutrient agar plates that lacked all the amino acids, and they lacked biotin and they lacked thymine. On one auxotrophic strain, they got no colonies. On the other auxotrophic strain, they also got no colonies. But when they mixed the two strains together, they ended up with a few bacterial colonies on this minimal media. These colonies are prototrophic. These colonies are now Met plus, Bio plus, and threonine plus, leucine plus, and thymine plus. So we combine the genetic information from this strain and this strain to produce prototrophic colonies. Circular DNA molecules are the molecule of choice in prokaryotic cells. Episomes are circular pieces of DNA that can exist either as extrachromosomal DNA elements or they can integrate into the chromosome of the host. They are a lot larger than plasmids, which are small, circular, sometimes infrequently linear, pieces of DNA that exist only as an extrachromal element. Plasmids never integrate into the host genome. They often confer advantage to cell resistance, to the cell for resistance to drugs and heavy metals. Okay, we're going to pay attention to the episomes first. F factors are episomes, and they have a lot of genes that are encoded in this circular piece of DNA. Specifically, they have a lot of genes that play a role in the transfer of the DNA. They're called Tra-A and Tra-B genes, followed by a capital letter. All these genes encode proteins that are necessary for the transfer of the genetic material, this F factor, from one cell to another. Conjugation takes place between two cells, an F plus strain and an F minus strain, and it's mediated by sex pili or F pili. Those proteins and this conjugation bridge is produced only by strains that are F plus, only by strains of bacteria that harbor an F episome. The pili act as attachment sites for the F minus bacteria. Those are the bacteria strains that do not have the F episome yet. Once the contact is made, the pili shorten, and a conjugation bridge is formed between the two cells, just like in the electron micrograph we saw a few slides back. Contact stimulates the donor cells to begin the transfer process. You're going to transfer a copy of the F episome into the recipient cell. The recipient cell is converted to an F plus cell, and the F plus or donor cell remains unchanged. Okay, let's look at some of the actual steps here. So we have a donor cell that contains an F factor, and we have a recipient cell that does not. So the F factor starts to make some of the proteins that are necessary for this conjugation bridge. And once this bridge has formed, then the transfer process can begin. There are a bunch of different proteins that are involved, something called a relaxosome, and a DNA strand that gets transformed into a piece of DNA that's competent to be transferred from one cell to another. The proteins of the relaxosome are released, and the DNA relaxase complex is recognized by the coupling factor and transferred to the exporter. So there's a lot of steps that are involved that are necessary to get one strand of this double-stranded F episome to attach to and begin the process of being exported into the other cell. The exporter pumps the DNA relaxase complex into the recipient cell, and then after the transfer is completed, both of these single strands become double-stranded. It's replicated in both cells, and now you have two strands that are F plus. They both contain an F episome. We're going to look at some permutations of that in the next talk. So in summary, genetic transfer can take place between bacterial cells by conjugation, transduction, and transformation. Conjugation takes place via a bridge or sex pili. F factors, or episomes, are extrachromosomal circular DNA that can exist in a bacterial cell. They are larger than plasmids and carry specialized genes to enable the genetic transfer. F episomes can also integrate into the host cell chromosome. Plasmids are circular pieces of DNA that exist only as extrasomal elements. Plasmids can confer advantages to cells, such as resistance to drugs or heavy metals. And cells carrying an F factor are called F+, and cells without it are F-. Transfer can only take place from an F+ to an F- cell. If a cell has an F factor, it cannot accept another one. Conjugation is the process of making the bridge and transferring one strand of the F episome into the F- cells. Cells with the F factor make the sex pili, which contracts into a conjugation bridge. The DNA is transferred through this bridge. At the end of conjugation, the F episome in both the donor and recipient cells is replicated to make it double-stranded.